All right, so what you should see when you open up Google Classroom so far for the Cell Anatomy Lab is the lab handout. Okay, that's the PDF document that I will be working from while I go over the pre-lab stuff, where all the questions and things are. Okay, there is a lab observations video, basically the video I took while I was doing the lab, so it's got pictures of all of the things we're going to be looking at. I will also show you that while we're doing the pre-lab. Okay, not the whole video, just kind of pictures. Okay, and then obviously your lab report, okay, with your name on it, that's what you should probably have open right now. Okay, so you can fill in a few things here as we go. Okay, so the purpose of the Cell Anatomy Lab is we've already had a lab where we got to just kind of use the microscopes and gain some experience with them. Now we need to look for specific parts of the cell. Okay, we know that the primary function of the cell is to carry out the basic functions of the organism. So what we want to do in this lab is look at cells from different kinds of organisms and see what they have in common. We already kind of know what they have in common because we've gone over the parts of the cell and we've looked at plant and animal cells, okay? But we kind of want to confirm that and see for ourselves, okay? What are the organelles, essentially, that are necessary for a cell to do the job it does? That is, to carry out the basic functions of the organism. That's the purpose of this, okay? So we're looking for specific parts of the cells. So that's our problem. Cells carry out the basic functions of an organism in order to survive. Thus, they must have structures in their cells that carry them out. We're going to figure out which ones. Okay, so in the design here, okay, we're looking at a bunch of cells, or we're looking at cells from different kinds of organisms. Okay, so we're not just looking at one type of cell, we're not just looking at one slide, we're looking at a few slides that contain cells from different types of organisms. We're going to look at a plant cell, we're going to look at an animal cell, and we're going to look at our own cells. Okay, we're going to look at all of those things today, okay, underneath the microscope. Right, what we're looking for when we look at those is common what? Common structures. Okay, we're looking for the common structures within all cells. Are we going to find and be able to see all of those? We're not. But if we can see at least some of them, then we've got a pretty good idea that, you know, this stuff is true. Okay. All right. Few things that we want to make sure we do. We want to look at all of these cells under high power magnification. Okay, we want to make sure of that so that we have a fair comparison between all of the organism's cells. Okay, you know, kind of same lighting and all of that kind of stuff. All right, for our hypothesis, okay, our if, and then statements. Okay, the if part should probably talk about what cells do. Spoiler alert, that's like this. Okay. The premise we're working on is that that's the job a cell does. So the premise is always the if part. Okay. The and part describes the experiment. In this case, we are looking at cells from different kinds of organisms. Okay. The then part needs to tell me what structures you think are going to be present. Okay. So I am looking for specific structures here, ones you think you're going to be able to see. Okay. If you don't see them, that doesn't necessarily mean your hypothesis is wrong. Okay, when you get to the conclusion, right? You can just as easily say many of the structures I listed were visible, these ones were not, here's the reasons why. Okay, it doesn't mean you have to reject your hypothesis if you don't see them. You can just accept it conditionally, okay, or something like that. All right, so you guys are going to, and we're going to have to walk down the hall and grab the microscopes because Mrs. Baxter needed to use the lab today for a chem lab, and I knew that we'd be able to look at the microscopes in here. So we're going to bring the microscopes in here, and we're going to use them in this classroom instead. So we'll have to go and get those, okay? You're going to get the necessary slides. They're over here, okay? So they'll be on like a desk here up front, okay? The things that we'll be looking at, right? We've got leaf cross-section, Okay, that's one of the slides we're going to look at. Okay, we're going to look at the animal cell, generalized animal cell. Okay, so what the leaf cross section is this. They've taken a leaf, you know, if you're looking at it like this, okay, what they've done is they've cut it this way, and you're looking at it straight on along its narrowest dimension. So what you're going to see on the microscope slide is this. You're going to see two leaves in cross section. One of those is what we call a monocot. The other is what we call a dicot. We want to look at the dicot, and it's usually the bottom one. All right. Now, the part of the leaf that we want to look at is here or here. Okay. We don't want to look at the middle. Okay. We want to look at the leaf kind of near the middle of the leaf, but not center of the leaf. Okay. The center of a lobe. Okay. So we're going to look at it there, okay, and we're going to see a bunch of plant cells. Okay, under the microscope. Even on high power, we're going to see a whole bunch of plant cells together, right? And then we want to do our best to label those. I'll show you what that looks like here under the microscope in just a minute. Okay, the animal cell is a piece of what we call stratified squamous epithelium. If you've ever dusted your knuckle, it's that red, painful skin that's underneath, okay, that takes forever to heal, right? That's stratified squamous epithelium. You will see just a square piece of that tissue planted under the cover slip, right? So we'll be looking at that. When you look at that, it's going to have, when you get it on high power, it's going to look like dry mud, okay? You know how when the water kind of dries up on top of the dirt, the mud kind of cracks, okay? That's kind of what this will look like. And again, I'll show you what it actually looks like here in a minute, right? It'll be pink, okay? So you'll kind of know if you're looking at it that way. And the last slide we're going to make is a wet mount of our own cheek cells, okay? We used to use a toothpick for this, but I had too many people who were drawing blood. And we can't have that because, well, that's gross. Blood can carry diseases like hepatitis and stuff. So we don't do anything that draws blood. So now we use a Q-tip instead of a toothpick. Toothpick works really good. Okay, so in order to make this wet mount, you're going to do like a CSI thing. Okay, you're going to take the cotton swab and you're just going to swab the inside of your mouth. Okay, give it a good wipe, and you're going to be twisting the Q-tip between your fingers as you go, so you get a good smear on there. Okay, and then you're going to do exactly the same thing onto the microscope slide. Just smear it real good. Again, spinning the Q-tip as you go. All right, and you'll see some goo on there. All right, that's your goo. All right, so then you'll put a drop of water on there and make a wet mount just like you did with the letter E. Your cells are going to be nearly transparent when you look at this. Okay, anything large, stringy, and dark is food. Okay, no matter how well you brushed your teeth or rinsed your mouth this morning, tiny little pieces of food remain. Most of them are fibrous, okay, especially if you have like toast or any bread products, okay, you're going to have that kind of stuff on there, okay? So if it's thick and dark, it is not a cell, all right? It is definitely something else, okay? Your cells are going to be quite a bit more transparent. Again, I'll show you what mine looked like when I did this so you get an idea of what you're looking for, okay? So drop of water on there, cover slip on, just like you did with the letter E. Everybody good with that? Okay. If there's time at the end, I have a prepared slide of human blood because everyone's always curious. What does blood look like? Can I prick my finger? No, you can't. Okay, because, well, we don't want to spread any diseases. So, okay, that'll be there if there's time. It is not part of the lab. There does not need to be a lab diagram of the blood. There does need to be a lab diagram of all the other cells. So I will also include the video on how to make the lab drawing again, okay, in this lab as well. Okay, so you're going to be taking pictures of all of these things under the high power magnification and labeling as much as you reasonably can label. All right, don't guess, don't make stuff up, okay? I'm looking for you to reasonably label as much as you can, right? So things you realistically should be able to see. If you can see the outside of a cell, you can see its membrane. Okay, if you can see where the cell begins and ends, that's the membrane. If you can see the nucleus, then you can see the nuclear membrane because you can see where it starts and ends. You should be able to see nucleolus, probably rough ER, okay? Lysosomes, chloroplasts, okay? If you're really good, you might even see mitochondria, okay? Occasionally, I have had people see Golgi apparatus, okay? You will not see smooth ER, It's completely transparent. You won't see it. Okay, you won't see ribosomes. They're too small. All right, things like that. Okay, but you should be able to see the majority of the kind of major structures. All right, so that's the kind of procedure part, okay? Looking at what you're gonna see. What you see right here, okay, this one here, this is the plant cell, okay? So this is the leaf cross section. What I want you to do is to be looking at the cells that are in this layer. Okay, it's what we call the palisade layer. It's the part of the leaf that carries out photosynthesis. The reason it carries out photosynthesis, what do you suppose these yellowish green things are? Those are chloroplasts. You'll see them that clearly. Okay, they're very evident. They're quite large. Okay, pink dots, those would probably be the nucleus. Okay, the little pink dot inside the nucleolus. You can see here on the edge, it looks like kind of has a beard. Okay, you're going to see all those things that I told you would see. Okay, now just to give you an idea here. Okay, like this is one plant cell. All right, you have to kind of look because the walls are, not as evident as they are in a hand-drawn diagram. Okay, but you can kind of see where cells begin and end. There'd be another one in between here, but it's pushed back so we can't see the structures that are within it. All right, so that's kind of what you're looking for. Everybody follow on that? Okay, for the animal cell, okay, what you can see in the animal cell. So is going to be something like this when you get it on high power. When you get it on like scanning and low power, okay, you'll see something like, well, this would be more on high power now that I've blown it up more, but okay, each one of these dots here is a nucleus of a cell, right? So you can see the cell membranes, okay? You can see stuff like that. You can see that each nucleus is kind of elongated. That's because one side has the rough ER on it. Okay, remember I said it looks like it has a goatee. Okay, you can kind of see that in most of the nuclei. Okay, things like that. So that's what you'll be looking at. If you need to use more than one cell to do your labeling, that's fine. Okay, you don't have to just label one particular cell. If you need to use the one cell to label the nucleus and one cell to label something else, that's fine. Okay, but that's what you'll see. Okay, blown up even larger. Okay, they can look like that as well, depending on how your slide is stained. There were two different stains used in the slides we have. This one's a little more faded, but again, here's a nucleus, a nucleolus. Okay, you can see kind of the cell membranes around them. Right, it's once you know what to look for, it's a little easier to find it. Okay. All right, for the cheek. Okay, these are both pictures that I took of my cheek cells. Right, so typically they're going to be a bit bleached out because they're not stained. Okay, no dye has been applied to your slide, so they're a little bit more transparent. One way to make them stand out is once you think you've got some cells focused, okay, you'll want to use the condenser diaphragm. That's this little tab right here. It will open and close the iris that is below the stage. Okay, just slowly moving that will make it get brighter and dimmer. As you dim it, sometimes the cells will just pop. Okay, it increases the contrast, the difference between light and dark, okay, or the edges of stuff, makes them stand out as you alter the amount of light that comes in. So that's a way to kind of make them pop out when you're looking at them. All right, so you can see here, okay, it's quite faded, but there's a nucleus. Okay, we've got like some mitochondria, these three dots here. Okay, there's some rough ER here on the edge. Okay, this over here is the Golgi apparatus. Okay, you have to. I'm not saying use your imagination, but use your imagination a little bit. Okay, again, be reasonable as best you can. Right, when you increase or change the contrast of the microscope, okay, you can sometimes make them stand out even more. Right, so again, there's cheek cells. There's a whole bunch of them there. Okay, you'll usually find them clustered. Okay, all right. So those are the things to be looking for, and then the blood looks like this, but again, that's only if we get to it. Okay, for the analysis for this lab, there's only one question, but I'm looking for a paragraph answer here. Okay, based on your knowledge of cell structure, so what we've gone over in class, and your observations of these cells, what organelles are most important for a cell to function? Okay, so I would say looking at what I saw, here's the here's the ones that were present in all the cells I saw. Here's what they do. Here's why they're important. Okay, and the second part of that reason, or the second part of that question is, which ones are specific to a certain mode of nutrition? So did I see something in one cell and I didn't see it in another? Here's what it does. It's related to being a photoautotroph or being a chemoheterotroph. Okay, use those words in your answer. Okay, so I'm looking for a paragraph here, right, for your analysis. That's the only analysis question. Right, your conclusion, copy paste your hypothesis based on what you saw, your observations, can you accept or reject it? And then sources of error. Okay, was there anything that gave you trouble? Okay, nature of your slides or whatever, okay, that could be fixed. Questions on what we're doing? All right.